And again, we also have the mitochondria. We have several of these little guys that are in here that generate ATP for the cell. We also have the Golgi apparatus, where proteins are sent from the endoplasmic reticulum to this particular structure in order to be packaged and sent out into the cell where they need to go. So those are the specific structures that make up a eukaryotic cell. And remember, these structures that contain membranes surrounding them, such as the Golgi complex, the mitochondria, these lysosomes, and then the endoplasmic reticulums, those are considered to be membrane-bound organelles. So kind of highlighted these attributes of the cell already, but now we're going to kind of look at a little more detail about the nucleus of a eukaryotic cell. And the nucleus is where all of the genetic material is stored. And by genetic material, in most eukaryotic organisms, we're talking about chromosomes. So you have chromosomes that are situated inside of the nucleus. You're also going to have nucleoplasm, which is what the chromosomes are situated in. And then that entire structure will be surrounded by a nuclear membrane. Let me try to blow up this image so I can get it to move. It's not, looks like it's not cooperating with me right now. But hopefully I can zoom in. There we go, just a little bit more. Okay, so in this image here, we have the kind of outline of what a eukaryotic nucleus looks like. One thing I didn't talk about was the fact that you also have nuclear pores that are embedded inside of the nuclear membrane, and it's through these nuclear pores that the DNA, after it's been replicated and has been converted into messenger RNA, again, another process of the central dogma theory that was known as, that we described as, that was referred to as the process of transcription. That messenger RNA will pass through the nuclear pore, and then it will make its way to the ribosome complex in order to be translated. So that process of transcription takes place here inside of the cell. And then you can see where the nuclear envelope is, which is essentially the membrane that surrounds the entire nucleus. Okay, so this is our typical structure of what a eukaryotic nucleus is going to look like. Again, you have DNA that's situated inside of the nucleus, and you also have nuclear pores that enable the genetic material to pass through the nucleus. So a lot of different structures that are involved in making sure that this particular structure is the key structure for a eukaryotic organism. One thing to note about the chromosomes are the site of where genes are located. And remember, genes make up every attribute of a specific organism. So an organism's complete collections of genes is going to be known as its genotype or its genome. So if you've ever wondered, you know, about kind of the genes that are your own body, such as where your blue eyes came from, or where your brown eyes or your green eyes came from, those attributes, I'm sure you know, were passed down through your parents. But they were passed down in the form of genes, and these genes were embedded or contained in the structure of the chromosome. And so those are the items that get passed on when cells undergo the process of mitosis, which allows for those genes to be transferred. And each gene is going to contain the information that's used to produce one or more gene products. So what happens is when that gene is encoded in the chromosome or your genetic material, and then the process of transcription happens where that gene is then converted from a DNA sequence into a messenger RNA sequence, then the messenger RNA will leave the nuclear pore and make its way to a ribosome, and then it follows the process of translation where it will be converted into a protein, and then that protein will be expressed by your cells. So hopefully all of that helps to tie back to our previous lecture when we were talking about the central dogma theory. So let's talk about the other genetic material that exists inside of the nucleus, which is that of RNA. Remember, RNA stands for ribosomal it stands for ribonucleic acid. So when we talk about messenger RNA, we're talking about messenger ribonucleic acid, which is essential for allowing the process of translation to take place, which translates our RNA into protein. Now, a lot of your genes are going to code for specific proteins, but we do have some genes that are going to contain codes that are going to be used to generate what we refer to as ribosomal ribonucleic acid and transfer ribonucleic acid. Both of these components are very crucial in the process of translation because ribosomal RNA make up the ribosomes in which the process of translation will take place. And remember, translation means you're converting messenger RNA into protein. And in order for that to take place, you have to have tRNAs, or transfer ribonucleic acids, to bring amino acids together to formulate a protein structure. So the key caveat here is that there's also RNA inside of the nucleus, and there's two types of specific RNA, ribosomal ribonucleic acid and then transfer ribonucleic acid, which are used both in the processes of translation. Now, the DNA that exists inside of a eukaryotic nucleus is going to, again, exist in the form of chromosomes, and those chromosomes will contain the genes that are used to express the proteins that a particular organism is going to have. And in humans, we simply have, in our diploid cells, we have 46 chromosomes, which means we have 23 pairs in total. And you're probably familiar with the standard X shape of a chromosome. When we talk about having 46 chromosomes, we're talking about having 1/2 of this X, but we consider this to be a pair of chromosomes. So this is one pair of chromosomes here. But in actuality, we have 2 chromosomes that exist. They're just linked up to make 1 chromosome in total. So our human diploid cells are going to have 23 pairs of chromosomes inside of the nucleus. And in terms of genes that are packaged inside these chromosomes, humans can have between 20,000 and 30,000 genes. Some of these genes we know what they code for, and some of the codes that exist we don't know what they code for yet. So we're still exploring those options, or exploring possibilities of what those genes are used for. Now, another a special component of a cell, both eukaryotic and prokaryotic cells, is going to be that of the cytoplasm. And this is a gelatinous matrix that is used to help allow catabolic and anabolic reactions to take place inside of it. But it's also a matrix that's used to help allow the organelles to situate themselves inside of it. And in the prokaryotic cell, we don't have any organelles, really very small. The only thing that's in the prokaryotic cells are the ribosomes, and then the nucleoid region. But all of that stuff is situated inside of the cytoplasm, which helps to maintain structure for the cell, to allow those structures to exist inside of it. The cytoplasm also contains storage granules as well as some organelles, and then all the organelles that we've talked about already, they all have a very specific function to them, so the cell doesn't have anything inside of it that it's not going to need. There's no aesthetic properties that an organelle would have that are purely for decoration purposes. Every structure inside a cell has a specific function to it. But what's important about the cytoplasm is that it consists of water primarily, and that water is used to absorb heat that's released when metabolic reactions take place. So the cytoplasm serves a very important component of, or a very important function in heat absorption of heat that's released whenever an anabolic or a catabolic reaction takes place. And remember, if you don't recall those reactions from the last couple of chapters that we've covered, catabolic reactions are when we are breaking apart molecules and releasing energy, and anabolic reactions are when we are building or synthesizing biomolecules and storing energy. Both require energy to take place, so energy is lost in both processes in the form of heat, and you want to kind of use the cytoplasm as an absorbing material to absorb that heat. So the endoplasmic reticulum and then the ribosomes are the next structures that we'll talk about. These particular structures, as I said before, are kind of our sites of where translation takes place. The endoplasmic reticulum is actually an extension of the nuclear membrane of the nucleus. There's a lot of membranes that the endoplasmic reticulum folds into, which creates kind of a transport network that's going to exist in the cytoplasm. So think of it as kind of like a highway in the cytoplasm. And then on the endoplasmic reticulum, you have these ribosomes, which are going to situate themselves on top of it. The rough endoplasmic reticulum will have ribosomes. The smooth endoplasmic reticulum does not contain ribosomes. And then remember, the ribosomes are made of that rRNA, the ribosomal RNA. And this is, again, where the process of translation takes place. And in translation, we have the generation of proteins. So where you see all these small little purple dots, those are all the ribosomes that are going to be used to generate proteins that will be used inside of the cell. So, and then also remember the smooth ER, that's where we have lipids being synthesized, but we don't use the ribosomes for that particular method. Ribosomes are pretty interesting in the sense that they kind of serve as these apparatus where you have a messenger RNA pass through them, and then as the messenger RNA is passing through it in the side of the complex, you have the translation of messenger RNA into amino acid sequences, which is going to give rise to a protein. And there's a key difference between prokaryotic ribosomes and eukaryotic ribosomes regarding their structure. And in prokaryotic ribosomes, you have what's called a 70S ribosome. And the 70S ribosome consists of a top portion, which is called 50S ribosomal subunit, and then also a 30S ribosomal subunit, which makes up the bottom half. And messenger RNA will pass through here, and you can have the process of translation. Now, in eukaryotic cells, we have a different ribosome, which is called 80S ribosome, and it will consist of a 60S subunit structure and a 40S subunit structure that come together. It's important to note the differences between these subunits because ribosomes are kind of a structure that we use whenever we want to kill a specific bacterial organism, because bacterial organisms have 50S, 30S subunits, but our cells, such as the cells of our gut, the cells of our skin, the cells of our esophagus, they contain these 60S, 40S subunits. So what we can do in the therapeutic world is we can design antibiotics that will target specifically the 70S ribosome structure. And an antibiotic will start to denature this 50S subunit or this 30S subunit, and that will prevent the process of translation from taking place. So that's why it's important for us to know the differences in the ribosomes between eukaryotic organisms and prokaryotic organisms, because if you have a prokaryotic organism harming a eukaryotic organism through infection, you can easily target these ribosome structures with an antibiotic to get rid of that prokaryotic organism, which is what we've been doing for a couple decades now. The Golgi complex, as I said before, is another structure that's used to allow essentially proteins being developed on the endoplasmic reticulum to be packaged and sent out to parts of the cell where they need to go. So you can kind of think of the Golgi complex as a mailing center. Items here get packaged and then they're sent out to where they belong in the cell.